So you may be asking, why utilize mammalian transient transfection for biotherapeutic protein production? Well, the answer is multifold. As drug targets become increasingly complex, proper protein folding, assembly, and post-translational modification becomes relevant. Performing your upstream steps in the same cell type, for example CHO instead of 293 cells, ensures that the product quality is similar to the end product and allows researchers to utilize the downstream processing expertise within their organization. In addition, preclinical drug discovery applications, there's a benefit to screening protein constructs quickly via transient transfection, which you can do in less than a week, while the stable cell lines are under development, which can take upwards of 3 months. And lastly, we're able to utilize transient transfection methods due to major advances in the field of recombinant protein production during the last decade. So this includes cell line engineering, optimization of expression vectors and serum-free media formulations, and also advances in the nucleic acid delivery methods. So today I'm going to focus on 3 important factors for influencing yield during transient transfection of suspension CHO cells. That includes growth media compatibility, cell density at the time of transfection, and lastly, the intrinsic properties of your protein. So there are many commercially available serum-free, chemically defined media formulations for the growth of suspension CHO cells. And it's known that highly anionic compounds contained in some of these formulations can severely impair transient transfection. So since the formulation of these medias is proprietary, in this experiment that I'm showing, we empirically tested 5 different commonly used media formulations for their ability to support transient transfection. So in this experiment, we utilized CHO cells and looked at antibody production from clarified supernatants at day 4 post-transfection. And we measured that using standard sandwich ELISA against the human FC region. So we're looking at mg per liter. And we utilized 3 transfection technologies for this experiment: the TransIT-Pro Transfection reagent from Mirus Bio, 25 kilodalton linear PEI from Polysciences, and the Freestyle Max transfection reagent from Life Technologies, now Thermo Fisher. So as I mentioned, we also looked at 5 different commercially available media formulations: 2 from Becton Dickinson, including the BD Select CD1000 medium and the BD Select CHO medium, Freestyle CHO expression media from Life Technologies, now Thermo Fisher, and 2 Lonza medias, the Power CHO 2 CD media and the Pro CHO 5 media. And hopefully you can see, looking across these bars, that there is varied expression levels depending upon both the media and also among different transfection methods. So just to walk you through this a little bit in more detail, so the best combination that we found was with the TransIT-Pro Transfection reagent and the Freestyle CHO expression media, with the BD Select CD1000 also supporting transfection pretty well with both the TransIT-Pro Transfection reagent and the Freestyle Max Transfection reagent. And also interestingly, with some of these other media formulations, we were able to get ample titers with the TransIT-Pro Transfection reagent, but it was barely above detection with either linear PEI or Freestyle Max. So this suggests that TransIT-Pro has better broad spectrum media compatibility, which can be advantageous when you're working with internal media formulations or medias that you are required to stay with. The next thing that I want to talk about today is the cell density at the time of transfection. So we found this to be a critical parameter for improving yield, specifically with our TransIT-Pro Transfection reagent. So in this experiment, we once again took CHO cells, spun them down and resuspended those cells at varying densities from 500,000 all the way up to 4 million. And then we added the same complex to those cells to minimize variability, and looked at antibody secretion from the clarified supernatants at day 3, 4, or 5 post-transfection, noted by the different colored bars on the graph. And hopefully you can see that there is a stepwise increase in your yield when you increase the cell density. It's about 8-fold, going from 2.5 mg per liter at 500,000 cells per mil all the way up to almost 20 mg per liter at 4 million cells per mil. So this was a really interesting finding to us. So we wanted to look closer. So we actually looked at both the transfection complex binding and uptake, and also the GFP efficiency at different cell densities. And we actually don't observe a difference in either of those parameters. So this suggests that just having more cells present that can be transfected with the TransIT-Pro transfection reagent at the time of transfection simply leads to an increase in the total protein that you can get out. So the last topic that I'm going to talk about today are the intrinsic properties of your protein. To address this topic, we took 5 different human IgG sequences from commercial products and had them codon optimized and synthesized in the same 2-vector backbone to express the antibody heavy chain and light chain. And so we compared the expression level of these 5 mAbs head-to-head using the TransIT-Pro transfection reagent, and once again just looked at antibody levels using a standard sandwich ELISA. So we're looking at mg per liter. And as hopefully you can see, we see varied titers depending on which of the 5 mAbs we're looking at. And it could be the most drastic difference we saw was between mAb1, which was at 8 mg per liter, and mAb3, which was at less than 2. So that can have up to, in this scenario, up to a 4-fold difference in your protein yield, depending solely on your antibody sequence, because these are all actually human IgG molecules. So in conclusion, transient transfection is an important tool for obtaining usable amounts of protein to better understand targets at an early stage. Protein yield is tightly linked to growth media formulation, transfection reagent selection, cell density and health at the time of transfection, and also the intrinsic properties of your protein. Optimization of these critical parameters will ensure experimental success. So I'd like to thank you for your attention, and I'd be happy to answer any questions that you may have. I've also included my direct email on this slide if you'd prefer to contact me that way. And we do offer free samples of our TransIT-Pro Transfection Kit upon request. So thank you.
So thank you. As a reminder, if you have any questions for Laura, you can type them into the chat window. Laura, so can you explain how does the transfection reagent formulation affect protein production?
So when you have a high efficiency and low toxicity reagent, that will allow a greater number of the cells to be transfected initially, which will lead to higher transgene expression levels and subsequently better protein yield.
And how does cell density affect protein yield?
So we were, honestly, we were quite surprised that it had such an effect on protein yield. And it really works well if you have a high efficiency transfection reagent that can bind and take DNA into a large percentage of your cells. Because by transfecting more cells at the time of transfection, you're increasing that total biomass for production. However, if you're growing your cells in batch culture, at high densities, they may crash sooner depending on the ability of your media to support that high density culture.
Okay. If you have follow-up questions for Laura, you can email her. Thank you for joining us today. Thank you, Laura.
Thank you.